Hi everyone, I'm Karthik, a senior interested in the field of bioinformatics and machine learning. For the past couple of years, I've done extensive research into the genomics field, seeing how we can use computational algorithms to make a real-world impact through projects, internships, and different graduate-level bioinformatics courses. Today I'll be discussing my research into how I use unsupervised machine learning methods to find clusters of related genetic variants that lead to the formation of breast cancer. Breast cancer is one of the most notable and deadly forms of cancer that are out there, afflicting over 2.3 million women, costing $40 billion in patient care, and causing 700,000 deaths annually. It often spawns from environmental and inherited factors in the human body, where the accumulation of cells metastasizes into other regions of the body. In this particular study, I focused on estrogen receptor positive breast cancer, or breast cancer that's caused by an overproduction of estrogen, estrogen receptor negative breast cancer, and general breast cancer overall. The current issue with breast cancer studies right now lies in the fact that genetic variations are currently studied in isolation and focus on a specific type of cancerous disease. Due to the abnormal growth of cancer, it can spawn in numerous tissues in the human body, leading to various different types of cancers, often called SNPs or SNPs or single nucleotide polymorphisms. These genetic variants are extremely hard to find due to the immensity of the human genome, as the computational processing power needed to run through this vast dataset is extremely taxing. Currently, there's misguided infrastructure and research allocation in these genomic studies, and a lack of collaborative effort to further this research. My research aims to fix these underlying issues by using unsupervised clustering to find specific related SNPs and their underlying genetic pathways. In essence, there are three main components. First, extracting the important genetic information from genome-wide association studies that tell a specific expression of specific SNPs on breast cancer, then clustering specific variants based on whether they have similar expression levels, and then mapping the variants that were clustered together into genetic pathways that can lead to the formation of breast cancer. First, to extract the necessary genetic variants, the MR-based library was used, a dataset that has numerous amounts of genome-wide association studies that comprise of billions of SNP associations. They tracked the sample size, the population that the study was tested on, the number of SNPs, and more. I was more interested in the beta value of the SNP, or the expression level that a particular SNP has on breast cancer. Principal component analysis, or dimensionality reduction, transforms this inputted data into a new coordinate system, where the number of axes was often less than the features present in the data. The axes of the principal components were ordered such that the first principal component has the greatest variance, the second has the second greatest variance, and so on. This reduces the number of dimensions that it's needed, while still having the information contained, and also allows for better computational runtime. All this stuff can be seen inside of my paper as well, the k-means, hierarchical clustering, and PCA. After I use PCA, I use k-means and hierarchical clustering to cluster different related SNPs together based on their expression levels. As shown on the table above, these different SNPs were then clustered based on different expression levels on the three different types of breast cancer, and afterwards these are mapped to specific genes through literature review and web scraping. Two main relationships are found. First, the MAPK signaling pathway was identified, which focused on 4 clustered associated SNPs, and this is seen on cluster 4 of the table. This signaling pathway is specifically vital for the proliferation of cells and is often changed by environmental factors. The second relationship is found with the connection between BRCA2 and ESR1, where uptakes in the ESR1 gene increase the probability of developing breast cancer, and is often seen to be correlated with BRCA1, which is known to be one of the leading susceptibility genes in breast cancer research. This is seen in cluster 2. The k-means algorithm had identified pertinent clusters that were shown to be expressive towards breast cancer through a more graphical visualization as well. Here are some of the principal components plots that I found that I plotted in order to find these specific clusters. Note that on the bottom graph, there are distinct clusters of red, gray, blue, and yellow, showing how k-means actually split up these different SNPs through computational analysis. However, on the other hand, the hierarchical clustering algorithm produced inconclusive results, as the clustering of these SNPs was disproportionate and didn't have a specific hierarchical structure, as many of these SNPs were grouped towards one section. Additionally, on the right, there wasn't really a definitive cluster size that could be shown through hierarchical clustering, as the score never reached a total minimum. The true power of this research lies in its general ability towards other types of cancers and other neurodegenerative diseases. I'm interested in focusing on other types of vital cancers like ovarian cancer, skin cancer, malignant melanoma, and perform the same pipeline to see how we can find other analysis as well. This dataset has low sample sizes currently for these cancers. However, through additional lab testing, we can increase the sample size of these datasets so we can increase the number for greater accuracy for machine learning models. Additionally, we can use other biological pathways discovered in the human body to further validate this through genetic testing. Currently, I'm also developing an open source platform so that researchers can run this pipeline by themselves on any particular type of cancer or risk factor. Thank you for listening, and I hope you consider me for the scholarship.